I'll start by talking about fatty acid. Really, this talk is going to be not necessarily related to injectable lipid emulsion, but I will talk more about fatty acid really independent of their presence in injectable lipid emulsion or ILEs. We will talk about the main differences between omega-3, omega-6, and omega-9 fatty acid. We'll talk about some of the mechanisms that are potential that might drive some of the effects of this fatty acid. And then briefly, we will touch on the composition on commonly utilized ILEs. So before we discuss the difference among fatty acids, I just wanted to clarify, and this might be obvious for many of you, that fatty acids are just a type of lipids, as you can see in this figure. And today we will focus our attention to fatty acid as well as triglycerides. And triglycerides that are both derived from diet and triglyceride derived from and lipids derived from endogenous triglycerides, which usually results in the release of lipid droplets when triglyceride undergo lipolysis. And I think it's very important to remember that the nutritional and metabolic effects of fatty acids and endogenous lipid droplets are really influenced by the kind and the proportion of the component of fatty acid. And today we'll talk a little more about differences between saturated versus unsaturated fatty acid. We won't talk too much about these in details, but as many of you might know, there are main differences in terms of the role of fatty acid and what in different states, and when either if we are in a fed state or in a fasting state. When we're in a fed state, so after a meal, for instance, usually triglycerides are transported through lipoproteins and through the adipose tissue and muscle lipoprotein lipase. Triglyceride can be broken down into free fatty acid, which are then reesterified into the adipose tissue, and glycerol, which can then be utilized for energy when needed. And within the skeletal muscle, also free fatty acid can be reesterified to triglyceride and stored as an intramuscular triglyceride. In a fasting state, then the adipose tissue plays a major role. As we know, triglyceride undergo lipolysis, and then the free fatty acid are released, as well as glycerol for utilization for the production of energy when, as needed. Obviously, we won't talk about all the different biochemistry and all the different pathways, but I just wanted to emphasize the importance of fatty acid and the beta oxidation. Fatty acid obviously needs to be oxidized for the production of energy, but really the beta oxidation is necessary to provide reducing equivalents to the oxidative phosphorylation, and so to allow eventually for the production of energy. And this occurs within the inner membrane of the mitochondria. Looking more about the structure, the chemical structure of fatty acid, and chemically, when a fatty acid is a carboxylic acid with an aliphatic chain or an aliphatic tail. And the predominant fatty acids are straight chain. They can be saturated, or they can contain double bond, carbon-carbon double bonds with an even number of carbon atoms. Fatty acid contain one or more double bonds in the chain. In that case, they're termed unsaturated fatty acid. When there is when the fatty acid contains only one double bond, we just talk about monounsaturated fatty acid. And when more than one, we talked about polyunsaturated fatty acid, or PUFA. In almost all naturally occurring UFA, unsaturated fatty acid, the double bonds are in the cis configuration. That means that the hydrogen atoms attached to the double bonds are on the same side. And usually they're positioned in the 3rd, 6th, or 9th carbon atom from the terminal methyl group. Now, if the hydrogen atoms are on the opposite side, the configuration is called trans. And so this is usually the result of food processing, in which, for instance, liquid oil were converted into solid fat. And so that process converts fatty acid from cis configuration to a trans configuration. Now, historically, there's always been a very clear distinction between omega-3, omega-6, omega-9, particularly with regards to inflammation, the effect on inflammation. In fact, omega-3 is always considered anti-inflammatory. Omega-6 have usually been considered pro-inflammatory, and omega-9, such as oleic acid, the main fatty acid found in olive oil, considered to be neutral from an immune standpoint. In reality, the evidence is not as strict and as strong as we might think. And hopefully today I'll convince you that really all these fatty acids, they have some immune properties. They can influence inflammation. And in addition to those, they will also talk about saturated fatty acid, which usually play a detrimental role in a number of cardiometabolic diseases. Here, I know that you will have access to this slide later on, so I won't go into the details of this slide, but I just put together some of the most common sources of different fatty acid, from MUFA to PUFA omega-3 and PUFA omega-6. Okay, so as I mentioned earlier, today we will focus on inflammation, particularly the effects that fatty acid may have on inflammation. And before we talk specifically about the fatty acid, I just wanted to introduce the concept of the NLRP3 inflammasome. The NLRP3 inflammasome is this macromolecular complex ubiquitary that is responsible for the production of pro-inflammatory cytokines, particularly interleukin-1 beta and interleukin-18 or IL-18. These pro-inflammatory cytokines are very well known to exert a number of detrimental effects. Particularly, they can induce insulin resistance, they can induce cardiac dysfunction, and I'll show you some data in a few minutes. And so really activating the NLRP3 inflammasome is considered to be detrimental in a number of cardiometabolic diseases. The inflammasome, as you can see here, requires two main steps: the priming of the inflammasome, so the formation of its components, and the triggering of the inflammasome, which will allow pro-caspase-1 to be converted into caspase-1. And this caspase-1 essentially cleaves pro-IL-1 beta and pro-IL-18 to mature IL-1 beta and IL-18, which are then released into circulation, and they can bind to their specific receptor, which are really found everywhere, including, for instance, the cardiomyocyte, and they can exert their detrimental effect. Now, over the years, we've always thought that inflammation would be more of associated with the severity of the disease or disease progression. However, we really never had the evidence to support the concept that inflammation might cause cardiometabolic diseases, particularly heart disease. Until only a few years ago, this is in 2017, the CANTOS study was published. The CANTOS study was a randomized controlled trial where patients with atherosclerotic cardiovascular disease were randomized to either placebo or canakinumab, which is an IL-1 beta monoclonal antibody. So it's a blocker of interleukin-1 beta. And what they found in this study is that patients who were treated with canakinumab presented a 15% relative risk reduction for the composite primary endpoint of cardiovascular death, non-fatal myocardial infarction, and non-fatal stroke. And you might be asking yourself now, why are we even talking about this study, since this was a lipid session? But the reason I wanted to show you this study is that this really proves that inflammation is not just a marker of disease severity, but is actually involved, it plays a causative role in the development of cardiovascular disease. With that in mind, in fact, earlier this year, only a couple of months ago, for the first time ever, the US Food and Drug Administration approved a drug called colchicine, which is being used for gout and other inflammatory disease for the treatment of cardiovascular disease. And so really now we know that inflammation is important because it is a mediator of cardiovascular disease. And so targeted inflammation therapies could be beneficial to prevent cardiometabolic diseases. Now, why I'm talking specifically about the NLRP3 inflammasome? Well, it's important to differentiate what is an inflammation mediated by, for instance, a microorganism. It could be due to an infection, and what is not due to an infection, what is called a sterile inflammation. And in this case, today we'll focus on the sterile inflammation that might be due to other components other than organisms, for instance, fatty acids. And do fatty acids influence the activation of the NLRP3 inflammasome, the production of interleukin-1, interleukin-18, therefore promoting the detrimental effect of this pro-inflammatory cytokine? And this was done in very elegant studies. This was a really pioneering study published over 10 years ago in Nature Immunology, where they looked at supernatants of resting or LPS-primed bone-derived macrophages, where they basically use LPS to prime the inflammasome. As I mentioned earlier, priming the inflammasome means to produce the component of the inflammasome, and then they provided palmitic acid, the most commonly found saturated fat in food. And they found that the more palmitic acid we give, the more interleukin-1 beta we produce. And same was for IL-18. So as you can see here, there is a dose-dependent increase in interleukin-1 beta and interleukin-18, suggesting a palmitic acid saturated fat can activate inflammation. There is also an increase in interleukin-6, but remember that interleukin-6 is downstream of interleukin-1 beta and upstream of C-reactive protein, which we measure clinically. So interleukin-6 would then downstream stimulate the production of C-reactive protein. In this study, they also show that if you inject intraperitoneally interleukin-1 beta to mice, and after 2 to 3 hours, you measure their glucose tolerance is impaired, as you can see here in the black box, suggesting that interleukin-1 beta really cause insulin resistance and glucose intolerance. However, when you use a mouse that does not have interleukin-1 beta, so it's a interleukin-1 beta knockout mouse, these detrimental effects are prevented, suggesting that really the negative effect of saturated fatty acid are mediated by these pro-inflammatory cytokines. As a group of researchers here at VCU, over the years, we have developed an orally available NLRP3 inflammasome inhibitor. So a medication that can block the NLRP3 inflammasome. And we've shown here in blue mice that are fed with a high saturated fatty acid diet, also known as a Western diet, present a worse cardiac function. So their cardiac function gets worse. This was measured with echocardiography. An example here at the bottom, you can see a reduction in left ventricular ejection fraction, a measure of systolic function in the mouse. And when we feed the same mice with the same diet, but we add this NLRP3 inflammasome inhibitor to the diet, you can see that their cardiac function is preserved. Again, confirming the detrimental effect of saturated fatty acid are heavily mediated by the NLRP3 inflammasome. Now, I hope I showed you some data and convinced you that saturated fat can increase the activation of the NLRP3 inflammasome. But could we inhibit now this activation using unsaturated fatty acids, which have been historically known as the healthy fats? So in this study, which was also another basic science study, but very elegant, they did really a similar experiment as before, where they prime cells with LPS to form the component of the inflammasome, and then they treated with saturated fat, 2 different ones in this case, palmitic acid and stearic acid, and you can see confirming an increase in interleukin-1 beta. However, when they treated the cells with monounsaturated fats, in this case was oleic acid, and polyunsaturated fat, in this case was actually an omega-6, so linoleic acid, they found that this activation of interleukin-1 beta was prevented, suggesting that these two fatty acids actually have an anti-inflammatory, we're able to prevent the increase in IL-1 beta and providing anti-inflammatory effects. They did an even more elegant experiment. They said, okay, instead of treating cells with just individual fatty acid, can we actually maybe inhibit or treat the inflammation caused by saturated fats? And so what they found here, they treated cells first with saturated fat, and then they added unsaturated fat, sort of what we would do with diet as a supplementation strategy. So we would add on top of saturated fat, the unsaturated fat. And they found that both oleic acid and linoleic acid, they were able to prevent the activation of interleukin-1 beta in the cells. So really, I think these were very powerful data to show that if you add this fatty acid on top of saturated fat, even if a saturated fat content might be elevated, we could potentially prevent the activation of the NLRP3 inflammasome and the production of pro-inflammatory cytokines. Now, this is another very important study, and this was published in Diabetes in 2015. There is, over the years, there's been this concept that adipose tissue, so fat mass, can be highly pro-inflammatory, but there's been evidence now to suggest that an individual could have an excess adipose tissue and present with overweight and obesity and still present with a relatively healthy metabolic phenotype. And so in this study, what they're trying to see is whether dietary fatty acid can actually influence the inflammatory, the inflammation within the adipose tissue itself. And they found that if you treat adipose tissue with monounsaturated fatty acid, particularly oleic acid, you do not increase the activation of the adipose tissue of interleukin-1 beta within the adipose tissue, even in the setting of obesity, while you could actually increase the IL-1 beta production if you just treat the cells with saturated fatty acid. Moreover, they looked at whether mice, even if they had overweight and obesity, they would still present with insulin resistance when fed with a high oleic acid diet. And surprisingly here, despite consuming similar amount of calories and despite being obese, these mice, they still had a preserved glucose tolerance when they were fed with a high oleic acid diet, suggesting that really, even if they provide the same amount of calories, 9 calories per gram, fatty acids present profound different effects on the immune system, even in the setting of obesity, of expansion of adipose tissue. This was another more recent work suggesting that the beneficial effect on monounsaturated fatty acid, particularly oleic acid, might not be just driven by their anti-inflammatory effects, but this is the work from Doug Mashek at the University of Minnesota, showing for the first time that monounsaturated fatty acid can allosterically activate the longevity molecule called sirtuin-1 or SIRT1, which we know can be activated by, for instance, resveratrol. And so now we finally have an additional activator of sirtuin-1, such as MUFA, monounsaturated fatty acid. So in this study, finally, hopefully I convinced you that monounsaturated fatty acid have profound anti-inflammatory effect, at least in basic science studies. Omega-6 might not be necessarily as bad as we thought. In fact, there are some data to suggest that they might even be associated with lower inflammation. But what about omega-3? Well, omega-3, as I mentioned earlier, have been historically known to be anti-inflammatory nutrients. And in fact, this has been confirmed in a number of studies. This was one of the first reports published in the New England Journal of Medicine from Dr. Charles DiNarello's group, who's the person who cloned IL-1 beta for the first time. And healthy subjects here received a large amount of omega-3. This was 18 grams of omega-3 per day. It was a mix of EPA and DHA. And you can see here that when these individuals received omega-3, high dose omega-3, their interleukin-1 beta level went down. After they stopped, 10 weeks after they stopped, the level remained low. But unfortunately, after 20 weeks from the end of the omega-3 treatment, the level of IL-1 beta went back up, suggesting that really this treatment should be chronic to exert their beneficial effect on inflammation. Basic science work also showed that some of the benefits of omega-3 might actually be mediated by the inhibition of the NLRP3 inflammasome, similar to what I showed you earlier with the other fatty acids. However, I showed you some, I hope, interesting, mostly preclinical data, but do fatty acid really, particularly unsaturated fatty acid, do really make a difference with regards to cardiometabolic outcomes in patients? And I think over the last 15 years, we've had 3 main studies that I really believe they changed the way we practice nutrition. The first one is the WHI, the Women's Health Initiative study. The second is the Predimed study done in Spain, and the CardioPrep study is also done in Spain. In the WHI, the investigators tested the hypothesis that a low-fat diet, which was being recommended by the American Heart Association and other societies over the last decades, would be associated with lower risk for cardiovascular disease. However, as you can see here, by the end of the study, this was a large randomized controlled trial. Low-fat diet did not cause any reduction in cardiovascular events. And so really this study killed, in a way, the field of low-fat diet for the prevention of cardiovascular disease because it really didn't work. But what about higher fat diet? So high unsaturated fatty acid diet. In the Predimed study, this was a primary prevention studies with patients who did not have an established cardiovascular disease, but presented risk factor for it. And the CardioPrep study was a secondary prevention study. So patients who had an established atherosclerotic cardiovascular disease. Patients were randomized to either in the Predimed to a low-fat diet, a high olive oil diet, or a high nuts diet, and in the CardioPrep, a high olive oil diet. And what they found at the end of the studies is that in both studies, this dietary interventions in a randomized controlled fashion showed a reduction in the cardiovascular events by 29% in the Predimed and about 27% in the CardioPrep study. Very powerful data. And really, when you looked at what changed in their diet, the main difference in this Predimed and CardioPrep compared to their baseline was an increase in extra virgin olive oil that was associated with greater improvement in cardiovascular events. Not only that, in the Predimed, there was no weight loss. So this is a very important concept, changing the idea that a diet only works if an individual is necessarily losing or gaining weight. In this case, we really did not see a weight change over time, and supports the concept that we can improve diet quality without necessarily changing body weight and still improve cardiometabolic outcomes. Other analysis of the Predimed, in fact, showed that even without weight change, there was a reduction in the diabetes incidence by 36%. There was a reduction in atrial fibrillation in patients who were receiving olive oil. And then there was also an analysis looking at the incidence of invasive breast cancer risk, showing that patients who were randomized to the extra virgin olive oil diet, a supplemented diet, they presented a significantly lower risk for being diagnosed with an invasive breast cancer. And also this was dose dependent. The higher was the amount of olive oil being consumed, the better were the outcomes. What about omega-3? Obviously, olive oil is not a main source of omega-3. In this study, we did a meta-analysis a few years ago where we confirmed that also omega-3, particularly the higher doses, are associated with the lower risk for cardiovascular disease. This is mainly driven by one study called the Reduce It, where they use a high-dose purified EPA, showing benefit on cardiovascular events. However, what we were a little surprised to see was that it was actually an increase for the risk of bleeding and an increase for the risk of atrial fibrillation in the studies, which was somewhat surprising, as we really didn't think that omega-3 could have any potential detrimental effects. And so we tried to explore this a little more. We did a follow-up meta-analysis, particularly focused on atrial fibrillation, and we did show that no matter what's the dose of the supplementation of omega-3 or fish oil, there was a significant increase in atrial fibrillation diagnosis in this across different studies, across different doses and different types. Some of the studies only use EPA, only DHA, and some was a mix. And so finally, now just one last slide, I just wanted to show you here the different composition of fatty acid of commonly utilized in injectable lipid emulsions. And with this, I wanted to just emphasize the importance of knowing the content of the different ILEs that we use. Often when we hear, for instance, fish oil containing ILEs, we think that all different ILEs, they have the same amount of fish oil. In reality, as you can see here, these are just two commercially available ILEs. You can see that the content of omega-3 is extremely different. And so if a study was conducted with one product, we should not extrapolate those results for the other product. Really, we need to be more careful at extrapolating data just because the study says that there are some some omega-3, for instance. And so I think this is really important to look at the different fatty acid content. And based on some of the data I showed you and what Dr. Junkier will present later, it really hopefully will help making some potential clinical decisions. And so in conclusion, I hope I convince you that fatty acids do have immunomodulatory effect, particularly saturated fatty acid, a negative effect by increasing inflammation, and unsaturated fat by inhibiting it. Targeting inflammatory pathways can prevent cardiometabolic disease. Particularly, UFA-enriched diets rich in olive oil and nuts can prevent the risk of cardiovascular disease and metabolic disease, particularly olive oil for metabolic disease. And PUFA omega-3 also prevent cardiovascular disease, particularly high-dose EPA. However, it is associated with a greater risk for atrial fibrillation when given as a supplement or as a drug, really.
Be sure to watch for upcoming Channel One videos on nutrition and learn more from our esteemed colleagues. Have a great day.